Hi, everyone. My name is Jeremy Wilkinson. I'm the global segment lead for microbial genomics here at PacBio, and I'll be the host and moderator of today's webinar. I want to welcome you all to this webinar with today's topic focused on the recent advances in unlocking human microbiome clarity with PacBio HiFi sequencing. And today we have two human microbiome talks from experts in the field. So just to start out with a little bit of housekeeping here. So we have a lot of great material to cover today, and the presentations portion of this webinar is pre-recorded, but it will be followed by a live Q&A session with all of the speakers. You are welcome to submit your questions at any point during this webinar by typing them in the area provided on your attendee control panel, and we'll get to them either in the chat, but likely at the end. Just a reminder that all attendees are muted. We also have uploaded a few pieces of literature to your control panel, so please feel free to download those. We will be recording this webinar and making it available shortly afterwards. Please keep an eye out for a follow-up email with the link to the recording. Immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill it out. It only takes a couple of minutes. It helps us understand your needs and plan for future webinars. And then you can stay up to date with upcoming webinars and events by visiting pacb.com/events. So again, this is a 1-hour webinar. We have talks and live Q&A at the end. For the Q&A, we have Dr. Dan Portick, staff bioinformatics scientist at PacBio that will be joining us. And like I said before, I'm joined today by two great experts, and the agenda is as follows. So I'll start out with just a brief introduction to HiFi microbiome sequencing and microbial genomics. And then Ashley Earl, who's the director of the bacterial genomics group at the Broad Institute, will talk about connecting the dots and recurrent urinary tract microbiomes. And then Jake Minich, who is a new assistant professor at Baylor University, will talk about culture-independent meta-pangenomics enabled by long-read sequencing. So with that, let's get started. All right, so I'm going to give a brief introduction to HiFi sequencing and specifically to microbial genomics and microbiome sequencing. So what PacBio HiFi sequencing is, they're sequence reads that are both long and highly accurate. So this is more complete and accurate long read sequencing. So with these highly accurate long reads, we can enable a broad range of applications for human, microbial, and plant and animal on both our Vega benchtop system, which is the one there on the left, and the Revio high throughput system, which is the one on the right. Of course, with the Revio system, you can do more of these applications at the same time or expand into these applications in the future as your laboratory needs grow. And then for microbial genomics, the applications can be metagenomics, microbial isolate assembly, epigenetics, viral sequencing, targeted sequencing, or full-length gene sequencing like 16S or ITS or 18S, for example. And so, like I said, HiFi reads really combine the best of two worlds. So this is high accuracy and long read lengths. So you get the accuracy that you would typically get with short reads, but at 100 times longer length. And so this is possible because we circularize the DNA in the library prep process using smart bell adapters. So these are hairpin adapters. And then we make multiple sequencing observations on the same stretch of DNA from a single molecule as the polymerase sequences around that, goes around that molecule. And so this washes out any random errors, which are shown as these little red dots in this graph, and allows for highly accurate consensus sequence to be called from those subreads. And this results in what we call a HiFi read that is typically an average of 99.9% or greater accuracy, or Q30. And the consensus sequence generation, so this HiFi reads generation actually happens on the instrument, so there's no need for additional compute off board for that HiFi regeneration. And if you want to explore HiFi data and see for yourself, we do have many data sets available. These are either on our GitHub page, PB Metagenomics Tools, or pacb.com/datasets. So these are all different types of microbial genomics data sets. And HiFi sequencing delivers more comprehensive and higher quality data for microbial genomics, and these are from culture to culture-free based approaches. So there are really four primary ways that folks utilize HiFi sequencing, plus a fifth, which I'll talk about next, which is viral sequencing. But this is first off microbial whole genome isolate sequencing. And so this is where you can obtain single conjugate chromosomes, so complete genomes, plus you get the complete plasmids as well, which we know plasmids are really important for inferring things like resistance. And you get consensus accuracies that are greater than Q40 and oftentimes greater than Q50. And you're able to also detect restriction modification motifs. So you can get the epigenetic and methylation information. With all of these things, you can do things like identify strains, antimicrobial resistance genes, and mobile vectors that may be mediating spread. And the important thing is short read polishing or hybrid-based assembly is not needed because you have high accuracy long reads. And then for all of these applications that I'm talking about here, they are at competitive costs with other technologies. And you'll see that in a couple of slides from now. The next application is full-length uh, gene sequencing for microbiome research. So this can be 16S for bacteria, ITS for fungal, 18S for microeukaryotes. You can also sequence full operons, rRNA operons, so like 16S, ITS, 23S, for example, for bacteria. And what this gives you compared to partial gene short read based approaches is you get more classified reads. Drastically more reads are being classified, and importantly, those are being classified down to species and strain level. And then for shotgun metagenomics, the first application is read-based profiling, which is mapping back to reference databases. So when you have these high accuracy long reads, this is really good at mapping back to reference databases. So this is because you have on average about 8 genes per sequence read, and you're able to get more and richer functional information. About 90% of the reads have at least one gene located within it, which gives you about, on average, 4 to 5 functional annotations per HiFi read. And you're also able to profile taxonomy with high precision and recall. And then for shotgun metagenome assembly, you're able to obtain many high quality metagenome-assembled genomes or MAGs, many of which are actually single contig, complete or near complete MAGs. And the accuracy really comes into play here. So it's not only long reads, but it's high accuracy, which is different compared to some other long reads, which helps you with resolving these MAGs at the strain level. So many of these MAGs are actually down to the strain level and not collapsed at the species level. The same thing goes as is with the microbial isolate sequencing. There's no reason to do hybrid-based assembly or short read polishing because you have high accuracy. And then lastly, HiFi sequencing gives you the ability to distinguish one virus within a crowd. And so there's lots of different ways that you can do viral sequencing with PacBio. You can use Amplicon-based approach. You can use target enrichment with hybrid capture. You can do full-length RNA sequencing, or you can use a metagenomics approach to capture everything that's within there and pull out all of the different viruses. And then the last slide here is these are all of the microbial genomics applications with the two PacBio HiFi systems. So the Vega system on the left and the Revio system on the right. These are just the recommended maximum number of samples per SMRT Cell. For the microbiome sample types here, these are based off the human gut microbiome. This also shows you estimated yield per sample and cost estimates per sample. So these cost estimates are based off the cost of library prep plus sequencing, so PacBio reagents for the whole genome applications, which are the first 3. And then the last 2, which are the Amplicon-based applications, this also includes the cost of PCR, so PCR library prep and sequencing. But what you can see is that the cost here that are highlighted in bold and magenta are very competitive of what it would cost to do with other technologies. And there are 3 different options for these whole genome sequencing applications, the first 3. This is because we have different library prep kits that are tailored towards your needs based off, scale, automation capabilities, so on and so forth. So we have our HiFi Plex Prep Kit 96. We have our HiFi Prep Kit 96. We have the Amplify protocol, which is really useful for ultra low input sample types, like swabs, for example, or water. And then we have Sequel Longplex kit, which is a PacBio compatible partner, which does the shearing and barcoding upstage, up front of the SMRTbell library prep with PacBio. And then for our 16S, we have standard 16S, which is just a single monomer of 16S or any amplicon in this case. And then we have Connects, which you'll hear about later, which just concatenates multiple of those 16S amplicons together to take full advantage of the HiFi sequencing, the long reads. And so when you have Connects, it just allows you to do more samples per SMRT Cell and get a higher read depth per sample than you can with the monomer-based approaches. That's it for me. Thanks for your time. And now we're going to move into the talks from our experts. Thanks.
Okay, thank you so much for this opportunity to talk about some of our research focused on recurrent urinary tract infections and how we've been using PacBio Kinnex technology to start to connect the dots. Okay, so UTIs, many of us are not strangers to this type of infection. In fact, it's predicted that at least 50% of women at some point in their lifetimes will have a UTI. But it turns out about 20 to 30% of those will go on to have something called recurrent urinary tract infections. You'll hear me call it rUTI. These are infections that can occur multiple times in the matter of months and can last sometimes for years. Obviously debilitating for the individual, but also, it's terrible for society. Turns out in the US alone, 15% of our antibiotics go to treating these infections, and we know that they're becoming increasingly difficult to treat due to antibiotic resistance. So it's a big problem. And yet, despite the issue, we know actually very little about the drivers of rUTI. So we've known for a long time that the gut has a role. So we are known to be our own reservoirs of our uropathogens. So there's linkage between the gut and the bladder. It turns out in some recent work with our great collaborators at WashU, team led by Scott Hultgren, we believe that the gut plays perhaps an even greater role in this, in potentially interacting the microbes in the gut, interacting with the host immune system, and potentially setting up susceptibility in the bladder. However, that work aside, we also have a great interest in sort of this organ that you see in the middle, the vagina. It's thought to also be a reservoir for uropathogens. And we have a number of questions focused on whether this organ with its microbes may also have a role in setting up susceptibility to rUTI. And so really that's the subject of the work I'm going to be telling you about today. So trying to understand whether the vaginal microbiota in particular play a role in susceptibility to recurrent UTI. So to get at this question, we have a study that we call the UPS study, the Uropathogen Surveillance Study. We enrolled a total of 70 individuals in this study, roughly split between two groups: controls, women who had one or no UTIs in their lifetime, and a set of rUTIs, individuals who've had at least 3 UTIs in the past year, leading up to enrollment. And so each of these women agreed to let us into their lives, answer lots of questions, but also provide lots of samples. And so each individual was part of our study for 24 weeks. And every 12 weeks, they come in, give us blood, urine, stool, as well as periurethral and vaginal swabs at each of the visits. And these women were also given at-home collection kits, so that should a time when a UTI occurred, they could provide us with samples at that time from the convenience of their home. And then they were asked to provide samples for 12 weeks weekly, so that we could follow them longitudinally over that time. Okay, so lots of samples. I think there were 300 or so visits in total, thousands of samples. And so what I want to focus on today are the vaginal swab samples. So this just gives you sort of a visual snapshot of what the sampling looked like. So on the left-hand side, we have our control participants. The x-axis is showing over time. So we have individuals giving us their 3 samples. But you'll notice on the right are our rUTI participants. A lot more sampling happening as we expected. These individuals were much more likely to have UTIs than controls. In fact, 17 of our 34 rUTI participants had a recurrence, and we captured 23 UTIs and lots of vaginal swabs throughout. So I'll talk a little bit about another journey that these vaginal swabs went down. But the first question we had was, are there differences in the vaginal microbiomes of these individuals, controls versus rUTI, that might give us some sense of what could be different and potential pointers to drivers? So that's a fine question, but a difficult one to answer using sort of tried and true shotgun metagenomic techniques. So it turns out when you just sequence the DNA coming from these vaginal swabs en masse, this is what it looks like. You get, for each of the 8 samples I'm showing you here, on the y-axis is showing the percentage of reads that map to the host, right? So these are really dominated by hosts, which makes it very challenging to look at the microbial communities. So this is where we partnered with PacBio very early in their development of the Kinnex platform, and it's been really quite an important system for us. And so just in sort of a quick snapshot, we took all of our vaginal swabs, put it through their Kinnex system, which enables us to take a peek at the full length 16S rRNA gene from our communities. And effectively, what the technology does is it allows you to really take advantage of the throughput, the high throughput of these sort of high volume new PacBio machines like the Revio, while also optimizing for the output and also the accuracy of these instruments. So effectively, the 16S Amplicons are generated, they're concatenated, and then run on these machines. So here's just some stats on the number of larger concatenated reads that we got from our full experiment. And then once we've done the informatic demultiplexing, the number of 16S Amplicons of about the expected length of 1.5 kb and really terrific quality. Okay, so what can we do with these data? So quite a few things. So I'll just start with looking at the species level data, the results of looking at these data sets. And so here we've organized the data such that each column here represents an individual, and sort of collating all of their data across all time points into one sort of snapshot per individual. And on the y-axis is the relative abundance of the various different bacterial species that we could detect using the Kinnex data. So I'm sure you can see there's lots of different colors. It seems that there are certain species that really predominate, which we expected to see. And if we look at these profiles through the lens of something called community state types, which is sort of a common method for the vaginal microbiome field, we can see that our individuals represented the 5 known sort of state types and roughly the abundances we would expect in sort of normal populations. So great. But of course, we wanted to know if these state types mattered in terms of rUTI susceptibility. And so here's the data organized in a way where we could start to take a peek at this question. And what we found is basically what your eyes are likely already telling you, which is when we organize these data in sort of the control bucket or the rUTI bucket, there really was no difference in the number of individuals who had any of these different community state types. So that didn't seem to be particularly explanatory or interesting. So one thing, however, that we know is that these CSTs can be dynamic over time in an individual. So here we have one of our rUTI participants over time, and we can see that, they start off with this blue CST1 and they change to this green CST3 and they come back. And we see that, the participant 5 is not unique, that this can happen quite often. And so we wondered whether maybe the CST type, whether there was any connection between that CST type and whether or not an individual has an active UTI. And so Joseph Intner, a fantastic computationalist in the group who wrangled a lot of the data and generated the results I'm going to be showing you today, did this analysis just asking, is the individual at higher risk of having a UTI given their community state type? And the answer was largely not really, didn't seem to matter a ton, although there was the CST1, which is typified by the species called L. crispatus that seems to be least associated with active UTI. And L. crispatus is actually quite interesting. And going back decades, there's been a lot of work thinking about this as a probiotic for rUTIs. It's been a bit of a mixed bag in terms of results, but it's interesting that we're seeing this in our data as well. But one of the real strengths, I think, of this approach is going even deeper, going beyond the species level and down to something that we call Amplicon sequence variants. So instead of a species, it's really kind of getting to the level of sort of a subspecies. And so here, what I'm showing is for each of the species that we can observe across our individuals, Joseph has now just delineated with these tick marks where a particular ASV begins and ends. And so you see, it gives us a lot more complexity in sort of drilling down in what these organisms really are. And so we've gone from now over about 300 species across all of our data sets to over 8,000 ASVs. And we're really super excited about diving into these data to kind of give us a sense of diversity over time and over space, different habitats in these individuals, since we have the urines, we've got the gut samples, we've got the periurethral swabs. But what I want to focus on right now is that we were excited to sort of take another shot at asking that question of whether there was something special about our rUTIs versus our controls, just in general. So that's these data here. And it turns out when you look at it this way, there are, in fact, a couple of Lactobacillus crispatus ASVs that seem to typify one group versus the other. So this ASV2 and to a bit lesser extent ASV5 seem to be enriched in our rUTI group as compared to our controls, which tend to have a bit more of this ASV3. And so that was pretty cool. And of course, we immediately want to sort of go to that next level of asking, well, what, does ASV2 and 5, do they matter? And if so, how? But it's a little limiting to sort of just take the ASVs at face value. You got to start to dig in a little deeper. And so what Joseph did was he went to public repositories. He grabbed all of the high quality Lactobacillus crispatus genomes that he could get his hands on. And then he assessed whether or not these different references, so each tip along this tree represents a different reference genome that he pulled from public repositories. And now he's annotating on this outside ring whether or not these references have any of these particular ASVs. So it turns out, like many bacterial species, Lactobacillus crispatus has more than one 16S sequence in its genome. And you can see that in general, these three sort of sit throughout the tree of life for L. crispatus and often co-occur, including, our sort of ASV2 and 5 of special interest. Okay, so that is interesting, but it doesn't really allow us to start to dive in easily into function, right? Because are we looking in this part of the tree or this part of the tree or this part of the tree? It's not clear at this point. So in sort of a tip of the hat to multi-omics, I want to share with you sort of the integration of a different data type that has started to give us some more power to dive in. So it turns out for every vaginal swab that we processed for the Kinnex system, we also had a companion vaginal swab that was used to plate or grow bacteria on plates in the laboratory. And so really the goal here was to be able to assess these vaginal communities for uropathogens. And so we strongly suspect uropathogens are present, but they're at very low abundance. And we suspected, and I think it is true, we missed them quite a lot, even in these deeply sequenced 16S profiles that we were generating. So these plates, what we do is we get everything that grows on these plates, we swipe all of the biomass off the plate, stick it in a tube, and extract the DNA. And in this case, we put it through sort of a more standard short-read metagenomic sequencing process to get at sort of these plate sweep metagenomes. And when we looked at those, here's data coming from these metagenomes. So each column similarly is coming from a single participant, in this case, in a single time point. And no, that's not right. They're coming from a single participant sort of aggregated together. And what we find is that, in fact, we see there are uropathogens. These are these darker gray species that are shown here. But we also see Lactobacillus crispatus. It turns out Lactobacillus crispatus is a facultative anaerobe, meaning that it can grow in the presence of oxygen and did so on these plates. And what was cool about that is it gave us an opportunity to dive a bit deeper. And so I just won't talk about it much, but we've over the years developed techniques that allow us to go from sort of a species level view within complex community samples down to strains. And so effectively, let's say in this example, we have this one sort of blue chunk of the bar. We can discern from it in this sort of cartoon example that there are for instance, 3 different strains of Lactobacillus crispatus. We can discern sort of relative abundances, and importantly, we can place them on the tree of life. And so, and then we can, of course, compare what we find in one sample to this, what we find in other samples. So, long story short, what the strange predicted sort of strains told us was that these rUTI-associated L. crispatus strains are actually coming from this single subclade within the tree. And so we're really excited about that. All right, so let me just quickly take us through hopefully the take-homes that I'm leaving you with today, including how fantastic the Kinnex system is. I mean, it's high throughput, high resolution, and it's really, really just a wonderful workhorse, particularly when you're dealing with communities that are, chock full of host. Based on these data, we've been able to glean some really interesting ASV level insights that we think are pointing in a direction between the association between certain lactobacilli and rUTI risk. A quick shout out again to integrating data. I think both, the data from the Kinnex were fantastic, and then bringing in this sort of plate sweep data together, powerful, but there are some blind spots, but together really helped us to zero on the specific subclade that we're very, very much looking forward to diving into further, so that we can begin to sort of really unpack what is going on at the gene and functional level that can hopefully help us understand why in the world this specific type of crispatus is thriving in this rUTI vaginal ecology, and potentially point us in directions for what might be driving rUTI susceptibility. So with that, lots of thanks. I want to first start with just the WashU team, an incredible set of collaborators led by Scott Holgren. I want to shout out Henry Schreiber and Karen Dotson. Lots of great funding over the years. I'm very humbled and lucky to work with an incredible team of folks at the Broad Institute. I want to point out Joseph Intner in particular, who drove the vast majority of the analyses that I talked about today. And last but certainly not least, I want to thank PacBio and Jeremy Wilkinson in particular, who's been a superior supporter of all of our work, and a wonderful person to be working with. All right, with that, thank you very much.
Awesome. Well, thank you all for coming and to this microbiome session and using HiFi. I'm super excited to share with you some of the results of our recent study. So, across the world, around 25% of kids, 0 to 5, are undernourished. And most of these are children which are short for their age or stunted. And wasting, which is a more severe phenotype, is a lower incidence. And there's a lot of reasons for this. So it's not just about getting sufficient nutrition, but it can also be access to clean water, hygiene, both macronutrients, micronutrients, the intersection of infectious disease, including parasites, malaria, educational status. And about 10 years ago, there was a really, really important study by the Gordon Lab at WashU showing that the microbiome actually plays a role and may even be partially causative of the most severe form of wasting. And to this day, most of the research has been focused on wasting, which is the more severe phenotype, whereas stunting has been less studied. And I would say that also for all of the microbiome research within this field, the absolute majority of it has used short-read sequencing, with most of it being short-read 16S sequencing, which is still the dominant methodology used in the field. 16S is great, but it only allows you to look at changes in abundance of different organisms and with a relatively low resolution. Shotgun metagenomics, primarily with short reads, allows you to get at a little bit more information, such as the gene content. But again, you're really restricted to looking at genes that go up or down in the system. And, with short reads, you can also, several new approaches have been trying to assemble the genome. So if you assemble the genomes, then you can look at the gene content within a given species or genera. And one of the challenges with short reads is that while you can get partial genomes, without having a complete genome, it's really difficult to actually compare and do pangenome analyses or comparative genomics of these microbes, because one or two genes missing could be really important. And in most of the cases, you could only have 10, 20, or even 30% of the genome missing. So long reads provides this really unique opportunity where we may be able to get complete genomes, which then could allow us to do comparative genomics and pangenome analyses in the context, in this case, of childhood undernutrition. So we wanted to apply this to a data set, but first we really needed to understand which sort of methodologies would be the best in terms of generating high quality and high numbers of complete metagenome-assembled genomes, or CMAGs, which I'll refer to. So in the beginning, this experiment, which really started about 2 years ago, our lab had been doing nanopore for probably the past 10 years. So we had a Promethean. So I ran all these samples through the Promethean methodology. And then we had also recently acquired a Revio, so processed through the Revio. And later on, then we also added the Illumina as a comparison, and specifically their synthetic long read approach with our collaborators at UCSD. Specifically, we looked at a cohort from Malawi, so sub-Saharan Africa over here. So this was from an interventional clinical trial that occurred back in 2015. And there were 2 villages in Malawi where participants were enrolled. And they, generally speaking, were about 1.5 years of age when they started. And then they were tracked for 11 months. And you can see that between these 2 villages, there are differing levels of malnutrition, so differing levels of acute malnutrition, different levels of stunting, different levels of environmental enteric dysfunction, which is diarrhea is kind of a phenotype of that, but it's a little bit more than that. It's inflammation of the small intestine. But we took 4 children from each village, so the fecal samples of these participants, and looked at a total of 6 time points, or 5 time points for one of them, across this entire study design. And then we did deep sequencing on different long reads. So I developed this high molecular weight, high throughput DNA extraction methodology, and which I can go into more if you're interested. We're currently writing a protocols paper on this, but as I mentioned, we processed these 8 participants in 6 time points, so 47 total samples through these varying technologies. So the SmartBell 3.0 kit using Megaruptor to shear, and then onto the PacBio Revio, a total of 14 runs, and then also using the Promethean with at the time the best or the highest accuracy base calling, and also with the R10.4.1 flow cells and the KIP14 chemistry, which was has the boosted accuracy. So we're using the best approaches available at the time for the technologies, and then we also ran this through a synthetic long read approach with Illumina. And our primary benchmark or endpoint we're looking at is the total numbers and the quality of the complete MAGs, in this way defined by completeness more than 90%, less than 5% contamination, and then a single contig. So immediately when we compare the top 100 longest contigs from these technologies, you can see that long read approaches or single molecule approaches, both PacBio and ONT, are clearly an order of magnitude higher than the short read approaches. And so from here, it's pretty clear that short reads is really a thing of the past, and long reads, you get way longer assemblies. And it was also clear that the PacBio approach generally across all the contigs has assembles into longer contigs. and that this was really specific, for the assembly method which was used. So when we looked at these 3 deep sequence samples and then did subsampling all the way down to 1 gig, this also becomes very clear in that the total number of complete mags, regardless of the assembly methodology, was always higher for PacBio than it was for ONT. And when you look at the results also vary within the assembly methodologies for each approach. So that's also important to keep in mind. But in terms of the amount of sequencing data to complete mags, the PacBio Revio generated 2.3x more Cmags than ONT. And then, of course, when we compare this and we put a dollar amount on this, so I basically calculated all of the costs up until this point, and this included DNA extraction, library prep, sequencing costs, and looked at the total amount of data input and then the total amount of mags or complete mags. We can come up with a dollar amount, and you can see again that the long read approaches even almost an order of magnitude cheaper than short read approaches. And our short read approach was actually generated 0 complete mags. I had to go out to another resource where they generated almost 10 terabytes of data. And you can see that, even with that, we're almost generating the same number of complete mags. And, between PacBio and Nanopore as well, the cost per mag or complete mag is much, basically cheaper with PacBio. So why is this? Why is PacBio generating more mags, more complete mags? So I think there's a few things. I think, in this case, this is, in terms of sequencing yield, while it wasn't significant, right, they were pretty similar in yields. Although I will say that our recent runs, right, with the Revio, I think our latest was our best was 80 or 85 gigs. So this has improved, especially with Spark chemistry. But the amount of usable reads, like Nanopore produces more reads. But when you look at the quality, so this is the accuracy of the reads, right? It's 2 orders of magnitude higher than ONT. And I think especially for microbial genomes, I mean, this is in my opinion, really the sweet spot of technology that I just don't know if it will ever be matched. But this level of quality really, really helps when you're trying to assemble very similar microbes or even strains or species within a complex community. The read lengths, median and 50 and whatnot was also larger for PacBio. Of course, ONT, you do get longer reads. If you're looking for these ultra long reads, you can get longer. But and that being said, when you look at the quality of the genomes, we can see that, and these are only in the complete mag comparison, but that both the completeness and contamination had a higher quality for PacBio as compared to ONT. And again, this is comparing best versus best. And then we also looked at the using this ideal approach developed by Nick Watson, that the genomes themselves were also of higher quality. So if you have an error introduced into your genome, then when you go to annotate it, the genes themselves could be truncated artificially. So if your genes are shorter in your annotated genome as compared to what's in the database, then you would see a difference here. And so that's essentially what you see in this plot to the right. And again, that also was significant. So why else might having full genomes be valuable to your data set? So we looked at this in the context of, we created this hybrid genome library or a genome atlas of the microbes in this data set, generating 986 complete Cmags. And we looked at the classification rate of, if you were to look at the sequence reads, when you just look the standard sour mash non-redundant database versus this database plus your custom database. And when you use a custom database, you can drastically increase the classification rate of your sequences. So without the custom database, over half of your data is unclassified, meaning you don't have any sort of taxonomic assignment. Whereas when you utilize genomes from your data in your data set, you can increase this classification rate, to 82% for a total of like 44 to 60%. So we applied this, broadly to the data set, and we found that the overall microbial diversity in children with poor linear growth was higher than it is with improving or good linear growth. This was also associated with breastfeeding status, and these associations were significant both for alpha diversity and also beta diversity, meaning compositional differences in the microbiomes. But we wanted to increase the sample sizes, so we sequenced at least around another 40 participants, 5 time points, so over 200 additional samples utilizing the SQL longplex methodology for the library prep, which allows you to do a very fast and efficient library prep using transposon-based enzymes. And we sequenced this across these pools across a total of 4 different Revio runs, and we also included one matched run with Revio versus Vega. And this was handled in collaboration with PacBio, and a special shout out to both Ian and Primo in the Apps Lab, and also a collaboration with SQL, specifically Joe Meller. And what this allowed us to do is to apply machine learning because we had a larger sample size, and we could classify the association, or we could classify whether a children had improving or worsening linear growth at 78%. And we think that, and hopeful that this will increase with additional samples. But what's really interesting is that when you have this strain level and even species level resolution, you can really start to discern differences in associations of individual, say, species with a growth outcome, or in this case, a nutrition outcome. So Prevotella, right, there's the most dominant genus in our data set. I think there's maybe 30 or 40 species. And what you can see is that it depends, right? Some of the species were associated with improving linear growth, whereas others were associated with poor linear growth. So this is really critical to have this level of resolution to be able to distinguish the importance between these various features or species or strains in your data set. So we also then, because we had these genomes, we could apply pangenome approaches, and specifically microbial GWAS. And this was performed at the genus level for these various, the top 10 abundant genera. And what we found is a total of 89 genetic associations with linear growth. Most of these were in the Prevotella, followed by Faecalibacterium, and then there were a few associations in Megasphere and Bifido. And of course, all of these targets need to be validated in future studies, but just this sort of approach was really not feasible without doing long-read sequencing. And in my opinion, is really exciting. So the other kind of final thing we did was look at genome stability. So because we had a time series of these participants, we could ask the question, well, how does the genomes of a given species change over time? So in this case, we would say we would look at the earliest time point, and then we would look at instances where that genome or that species was found in at least 2 additional time points. So that had to have at least 3 time points from which it was found. And then we could compare the genome similarity of these latter time points to the initial time point. And we looked at this in these 8 participants. So again, 4 had improving linear growth, 4 had worsening linear growth. And what we found, which was really interesting, is that children with poor linear growth had a higher change or differences in the genome average nucleotide identity over time. And this suggests that either the genome species are evolving or they're being replaced by similar organisms. And it suggests stability or instability in the microbial genomes as a association or an indicator of gut health or linear growth status. So in conclusion, undernutrition is really important globally. It needs more funding, it needs more attention. We still really don't understand what are the exact causes from the microbiome, but we know that stunting is very high in Malawi, environmental enteric dysfunction also very high. So we generated a really unique data set using varying technologies. We show that long reads is superior, with PacBio being the most efficient way to generate complete mags. We then apply this approach to our long read data set and specifically show how across time, strain diversity, stability, and overall diversity was associated with linear growth status. We also performed pangenome analyses of microbial GWAS from these genomes. And we also show how genome stability could be a new way of understanding gut health, which is really only feasible using long reads. So we're happy to report that just last week, this finally came out in Cell, and there's a really nice write-up from the PR team at Salk. I'd encourage you, if you're interested, to read more about it. All the data is public. It's always been public. And finally, I've just started my lab at Baylor, and this picture was taken today. This is the day that I got my Vega, so I was pretty stoked. And in any case, I haven't found a proper hat for the Vega or a name yet, but if you're interested, I'm hiring PhD students, postdocs, and what I'm really interested in next steps is, can we use this microbiome to develop predictive diagnostics of future health outcomes? I'm really interested to understand more of the mechanisms of the microbiome and looking at other types of features, metabolomics, proteomics. And then I'm also very interested in applying this to agricultural systems, specifically aquaculture, fish farming related systems. How can we improve animal health, animal production with the microbiome? So with that, really like to thank Todd Michael at the Salk, my collaborators, Mark Mannery and Kevin Stevenson at WashU, Mike Tiza from Baylor College of Medicine, Rob Nayan Omar, and then some of his other trainees at UCSD, and then various folks at PacBio and Sequel. And with that, I can take any questions. Thank you.
Great. Thanks for the great talks. Thanks for those that have stuck around. We do have about 10 minutes for question and answers. Just a reminder, as you leave, do complete that survey if you would. It only takes a couple minutes and really helps us for planning for the future. So first question that we got, and please, if you have questions, please add them to the question panel in the question area there, and we'll get to them if we can. If not, we will try to follow up via email for any unanswered questions. So first question is for Ashley. So first off, some congratulations here, Ashley. Wonderful talk and work. This is really exciting to see. I'm curious about your plate sweep metagenomics approach, given that you're selecting for just cultural or culturable organisms, and specifically those that might outcompete some slower or more finicky growers on your media. How do you account for the bias that this introduces and integrate those findings with the metagenomics sequencing first approach to make sure you're looking at all of the potentially implicated species? It's a long one.
Yeah, great. No, it's great. And Sushmita, hi Sushmita. I know Sushmita. Thanks for the kind words and the great question. Yeah, so I think for this sample type, we have to be careful how we use these data, and it has to, the question has to match that bias, so to speak. So here, and we were using it in this case a bit in more of a confirmatory path. So we knew that we had this 16S signature, and we, I guess, were expecting to find things on the plate based on our strain tracking that would sort of recapitulate that signature. I think that is what we found, but I think importantly here, it allowed us to kind of hone in. Now, having said that, I think the point that you're raising, which is, this is not looking at the native community. So this is selected, it's absolutely biased, and we knew that going in. So, I think it is worth, if I'm wearing the reviewer's cap, some extra care to go in and verify using sort of the native DNA from that sample, just to verify some of our strain analysis. So I think it's a powerful orthogonal view, but you're right, we have to sort of use it appropriately. And we're thinking about using these data for other things, and we are wrestling with should we just be using absence presence? Should we be taking into account relative abundance at all? So it's active research for sure. Thanks for the question.
Great, thanks, Ashley, and thanks for the question. Please remember to put the questions in the chat if you have them. We have another question, which I can try to answer. Has nothing to do with the really interesting presentation. So again, kudos to you two, but interested in using PacBio instead of bacterial PCR. In particular, wondering how many reads I need to calculate in order to achieve comparable sensitivities, and whether anyone has experience with DNA extraction for this purpose. Would a metagenomics approach with connects due to high host DNA content be more appropriate, or a 16S approach? So I'm not super familiar with that. Is anyone else on the here on the panel familiar with bacterial work? Maybe Ashley or Jake?
Bacteria, I mean, think about the tree of life. There's the bacteria, the archaea, and then the eucs. So I think the eubacteria are part of the bacterial part of the tree. Yeah. I might be extending my neck out here. If so, then you're cool. Definitely, connects would be a great approach to apply in scenarios where you've got a lot of host content. Yeah, it should work. I hope I'm not making a fool of myself. I'm only a microbiologist for many decades.
No, that's what I assumed as well. So yeah, I think that if it is heavy host, then connects or just 16S in general is likely the best approach. You can do metagenomics, and then you asked about DNA extractions for if it's heavy host. I have seen some success with the QIAMP extraction kits from QIAGEN, so that's one route that you can take to try that out. And then you may be able to do a shotgun metagenomics approach depending on how that looks on a pilot run for host contamination.
And just quick follow-up, Wiki says Eubacterium is a genus of gram-positive bacteria in the family Eubacteriaceae. So yeah, connect should be great.
I have a question for you, Jake. So congrats again on getting the Vega in your own lab and starting your lab up. Do you have any cool projects already planned to use on the Vega?
Yeah, I mean, a few. So we're doing a project on rainbow trout. So looking at microbes and growth in trout with, it's a big producer in Idaho. We got another project which is pretty rad. So we have bears are our mascot here, and I think Baylor is one of the only zoos for universities. But anyways, we have two bears and two girls, and they're somewhat young, and they have very good dietary records. So I'm really interested in diving into dietary readouts, which is a bit different than microbial ecology, but it's the same sort of approach. And trying to, eventually also use that for human health, because dietary surveys are notoriously bad, but also for health of the bears and that sort of thing.
Cool. So is that going to be collecting fecal samples, doing time series, and
yes,
with the dietary? Have they already been collecting fecal samples for a long time, or is this just starting?
Just starting, yeah. So unfortunately, I don't have them as babies, but or cubs. But yeah, it'll be a really nice, rich data set over time.
Is that what your students and postdocs would be doing, collecting the bear samples? Actually, Jake, do you mind? I have a question for you. So now that you mentioned using the Revios, you now have a Vega in your shop. Do you have sort of like a rule of thumb in terms of like the number of samples, the kinds of samples like you would need to have to like push you towards one of the instruments over another? Have you started thinking through that for your own work?
You mean like to get a Revio or?
Yeah, I'm assuming you have access to one. That might be incorrect, but let's pretend you had a Revio and you have now this Vega. Like, how are you thinking about using them? How would you think about it?
Yeah, I think that's a good question. I see it as different from the Broad, but I mean, I see there's a great. I want to eventually integrate it with teaching and stuff as well, or at least. But if it starts to really scale, then I could see getting additional Vegas. But the Revios, you got to run 4 plates at a time, and unless you have like a good chunk of funding, and you really have those samples. I mean, a lot of places, right? It can be hard to keep them fed, and I think that the 1 plate thing, obviously, it's lower throughput if you're doing lots of samples, but it's kind of optimal, right? To kind of have one run at a time, similar like to the MySeq. So I see a lot of parallels with that, and I think, maybe the throughput's not as high, but I think that ability to be able to have consistent generating data could be quite valuable. Yeah.
Cool. All right, one last question here before we break off here. But Jake, you got another question here from Hannah. Hi, Hannah. If we extrapolate the results of your study, do you think microbiome interventions, for example, diet or probiotics, can decrease stunning in low and middle income countries?
Yeah, it's a great question. So, and that's a lot of like Jeff Gordon's work. They're working on these like microbiome based therapeutics. I think it's an interesting concept. I spent a lot of time in these places, and I think that there may be other approaches that might be a little more scalable. I think anytime. This is like a philosophical thing between prevention and therapeutics. I think that we really need to be focused on prevention, and that's kind of where I'm trying to focus on. Can we, have a predictive diagnostics? Can we identify areas at risk and then try to intervene earlier? That's assuming that you even have interventions, which is not always the case, right? This is another big challenge, but yeah, it could be, but then you got to think about delivery, right? You have to think about cold storage, like all the same problems we have with vaccines. It's not that straightforward, even if it works. So
totally. You got someone that agreed with you too in the chat, so that's good. All right. Well, we are at time, so just to be conscious of everyone's time. But again, thanks to all of you for attending, and thanks to the speakers for your wonderful talks and joining us today. Please keep us in mind for future webinars. Hope to see you at future events as well. Thank you all.
Thanks, everybody.
Thank you.